Biological research conducted on the effects of scalp cooling has focused on the hypothesis that cooling reduces the entry of chemotherapy drugs into human keratinocytes. Research has confirmed that cooling can reduce cytotoxic drug uptake by a minimum of 4-fold, and this can increase up to 8-fold depending on the temperature, due to a number of possible mechanisms which occur as a result of scalp cooling. Understanding these cellular mechanisms, which work in conjunction to be cytoprotective, allows us to both predict which chemotherapy regimens scalp cooling will be less effective for and find solutions to increase its efficacy. The first of these mechanisms is vasoconstriction. Scalp cooling triggers vasoconstriction, resulting in a reduction in blood perfusion to between 20 and 40% of normal blood flow levels. This reduces the extent of drug exposure and thereby reduces hair follicle damage overall. The second biological mechanism is a reduced drug uptake into the hair follicle cell. The low temperature of the scalp decreases the rate of drug uptake across the cell's plasma membrane due to a reduction in both active transport and diffusion of the cytotoxic drug. Chemotherapy drugs, such as the anthracycline doxorubicin, enter the cell membrane by active transport via membrane proteins as well as passive diffusion. However, low temperatures can cause a significant reduction in the kinetic process of active transport. Hypothermia also reduces drug uptake, as it impacts the structure of a cell's membrane fluidity. Lowering the temperature causes structural changes to the plasma membrane, thereby dramatically reducing its permeability for chemotherapy drugs. The third mechanism is a reduction in the rate of hair follicle cell division. During the active phase of hair follicle growth, the cells are continually stimulated by growth factors, so are thus in a highly proliferative state. As a consequence, these cells are more vulnerable to the cytotoxic effect of chemotherapy drugs. Research has found that because cooling reduces the metabolic activity of cells and the rate of cell division, this renders these hair follicle cells less susceptible to being targeted by the chemotherapy drugs. This is particularly important for chemotherapy drugs that target specific phases of the cell cycle, such as taxanes, which are mitosis-targeting microtubule destructive drugs. The final biological mechanism of scalp cooling is the reduction of metabolic activity. There are a myriad of cellular biological reactions occurring within cells, including metabolic activity, cell growth and cell death, all of which are acutely temperature dependent. So, when exposed to the effects of cooling the scalp, the reactions begin to decelerate a range of cellular processes, including oxidation. Oxidation is a process that is involved in chemotherapy drug-mediated cytotoxicity. By reducing the generation of cellular reactive oxygen species, scalp cooling may attenuate toxicity and hair follicle damage by reducing the occurrence of oxidative damage and ultimately cellular destruction. Scalp cooling can be a powerful cytoprotective tool because, unlike other methods to protect the hair follicles from cytotoxic drugs, it can mediate the damaging effects via all of these concurring mechanisms.